Hi everyone, welcome to the video lecture for Unit 2, Module 3.2.2. This one we're going to be talking about platelets. It's rather short, so just hang with me for a minute. As always, here are our learning objectives. So in a blood smear, platelets appear as spindle-shaped cell fragments. They average about 4 micrometers in diameter and are roughly 1 micrometer thick. Platelets in non-mammalian vertebrates are nucleated cells that are called thrombocytes. In humans, they're cell fragments rather than individual cells, so the term platelet is referred or preferred when referring to our blood. Platelets play a major role in vascular clotting systems and also include plasma proteins and the cells and tissues of the blood vessels. Platelets are continuously replaced. Each platelet circulates for about 9 to 12 days before being removed by phagocytes, mainly in the spleen. Each microliter of circulating blood contains about 150,000 to 500,000 platelets. The average concentration is about 350,000 per microliter. About 1/3 of the platelets in the body at any moment are in the spleen and other vascular organs, rather than in the bloodstream. These reserves are mobilized during a circulatory crisis, such as severe bleeding. An abnormally low platelet count, roughly around 80,000 micrometers or less, is known as thrombocytopenia. Thrombocytopenia generally indicates excessive platelet destruction or inadequate platelet production. Signs include bleeding along the digestive tract, within the skin, or occasionally inside the CSS. In thrombocytosis, platelet counts can exceed 1 million per microliter, and thrombocytosis usually results from accelerated platelet formation in response to infection, inflammation, or cancer. So the functions of platelets include releasing chemicals important to the clotting process, and by releasing enzymes and other factors in the appropriate times, platelets help initiate and control the clotting process. And they are forming a temporary patch in the walls of damaged blood vessels, and platelets can clump together at an injury site that form a platelet plug, which can slow blood loss and clotting takes place. And then finally, reducing the size of a break in the vessel wall. Platelets contain filaments of actin and myosin, and therefore, after a blood clot has formed, platelet filaments contract to shrink the clot and reduce the size of the break in the vessel wall. So platelet production, or thrombocytosis, takes place in the red bone marrow. Normal red bone marrow contains megakaryocytes with large nuclei. During the development and growth, megakaryocytes manufacture structural proteins, enzymes, and membranes. They then begin shedding cytoplasm in small membrane-enclosed packets. These packets are the platelets that enter the bloodstream. A mature megakaryocyte gradually loses all of its cytoplasm, producing about 4,000 platelets before phagocytes engulf its nucleus for breakdown and recycling. 3 substances influence the rate of megakaryocyte activity and platelet formation. They are thrombopoietin, or TPO, or thrombocyte stimulating factor, which is a peptide hormone produced in the kidneys, and maybe other sites, but science isn't too sure about that one, that accelerates platelet formation and stimulates the production of megakaryocytes. 2, interleukin 6, or IL-6, which is a hormone that stimulates platelet formation. And 3, multi-CSF, which we talked about in the last module, which stimulates platelet production by promoting the formation and growth of megakaryocytes.